Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Delphi Panels, What Are They Good For? In this webinar, three experts will take a deep dive into Delphi panel methods, their implementation, and provide pharmaceutical industry insights on the panel's uses and outcomes. I'm Sydney Otamanchak from the events team at scientist.com, and I'm happy to be your host for today. I'd also like to thank the sponsor of today's event, Partnership for Health Analytic Research, PHAR, for making it all possible. I want to take a moment now to introduce our speakers for today's session. Our moderator for today is Dr. Michael Broder, the President of Partnership for Health Analytic Research. Next up, we have Kathleen Villa, the Executive Director of Medical Evidence Generation in Medical Affairs at Ionis Pharmaceuticals. Our third and final panelist with us today is Dr. Tiffany Kwok, the Vice President of Health Economics and Outcomes Research with Crinetics Pharmaceuticals. And without any further delay, I'm going to welcome Michael. Good morning. Take it away.
Good morning. Thank you, Sydney, and thank you all for joining us this morning or afternoon or evening, or depending on where you are. What we're going to do over the next hour is take a look at Delphi panels and the variety of uses that they have. And just in terms of structure, what I want to do is kind of introduce myself a little bit, tell you a little bit about my company, PHAR, and then I'll talk about, I'll try to give you a high level overview of what is a Delphi panel, what's in my head when I use that expression. And then we'll zoom out a little bit and talk about the multitude of different methods that can be used to get at things similar to what a Delphi panel does, ways of gathering expert opinion. And then I'll look at some differences between those methods and tell you why the modified Delphi method has become my favorite. And then that'll take maybe 10 or 15 minutes for that background. And then I'll bring on the panelists and we'll have a discussion. We'll spend most of the rest of the time talking about their experiences using the modified Delphi panel method in their work. And then we'll leave 10 or 15 minutes at the end to answer questions. But I want this to be as useful as possible. So if, as I'm going along, there are things that are not clear, just go ahead and type the question into the chat and or into the. I'm sorry that your chat is disabled, into the Q&A section, and we will try to get as many of those questions as possible. And if we can't answer them all during the presentation, we'll try to do it in follow-up after the presentation is over. So. Welcome. That was this. So my background is that I'm a clinician by training, an OBGYN really, who worked in academic practice at UCLA and at the Rand Corporation for a number of years. But a little more than 20 years ago, I left to start the Partnership for Health Analytic Research with the idea that bringing together the kind of clinical experience and methodologic training that I had gotten during my training and fellowship with people with a lot of experience in the pharmaceutical industry, we could provide health economics and outcomes research in a way that really was different from what some other groups were doing. And we've been at it for 20 years now and have quite a bit of publication track record to show that the work that we're doing is of scientific merit. PHAR as a company is a little more than a dozen researchers. And yet we have this, more than 800 publications in 300 different forums over the years. And most of our work is in real world data analysis. So we probably 70% of those publications are using various retrospective data, secondary data sets to do retrospective studies. But a good 10% of it is using expert panel methods. And I've been using these methods since I was at Rand, where I kind of learned how to do it. And today I hope to share with you why I find the method so useful. First things first, as I said at the outset, I want to kind of give you a mental picture of what I mean when I say modified Delphi panel. This is a little cartoon. It breaks the modified Delphi panel process down into 4 or 5 steps. Obviously, it's a lot more complicated than this, and we'll talk about through the course of the hour, talk about some of these elements in a little more detail. The big picture, the way it works is we start with a question. And for the modified Delphi panels that we do, that question is usually of a clinical nature. Delphi panels can be used and are commonly used to estimate probabilities, numbers of things, and output like that. But for us, usually there's a clinical question. So a real example we had was that we did a panel to determine how often people with sickle cell disease should be seen in regular follow-up. And that depends, of course, on how sick they are and medications they're using, et cetera. And so, but that was the question. And so how to answer that question, the first step is review the literature and develop this evidence summary on what scientific information is there on how often people should be seen. Then we do what we call developing a rating form. And a rating form is really nothing more than a detailed survey with a series of patient scenarios in it. And so in the example that I'm giving you, those scenarios would be something like, a patient who has sickle cell disease with mild to moderate end organ damage, who's had 2 unscheduled visits in the last year, and some other characteristic. How often should this person be seen by their provider? And then another scenario will be just slightly different from that. Somebody with severe disease with those otherwise the same characteristics. And we take our experts, and for us, that's usually a dozen or so experts, and we give them that survey. And they answer all of those questions in advance of ever meeting each other together. So that's what we call the first round pre-meeting survey. Then we bring those panelists together in person, ideally, but we've been doing it, as you can imagine, virtually for the last few years, and it's working just fine. But I like in person better. We bring them together in person, and then we have a discussion of areas where they agree and disagree. So we've taken all their individual ratings and combined them in such a way that we can see, here's a question where, one person said that this type of scenario, the patient should come back in 3 weeks, and another person says 3 months. So in that in-person discussion, what the moderator does is guides people to share their opinions. We don't fight about things. We don't argue. We just share opinions. I think, we should see this person and follow up in 3 weeks because they're pretty sick. Somebody else says, no, I think 3 months is enough because whatever. So that's sort of the in-person discussion. And that's usually a full day process. At the end of that process, all the panelists do those ratings a second time. So they get a new blank rating form and they fill out their ratings. And what happens is, after hearing the discussion and thinking through some of these questions from somebody else's perspective, panelists will change their opinion and they will give different answers. And so we take the second round ratings and we analyze those. And where there's agreement on those, that's what we call the areas of consensus. So high level, that is the Delphi panel process. That's actually what I would call the modified Delphi panel process, I should say. The classical Delphi eliminates that in-person meeting. And we usually do 2 rounds of ratings with a meeting in between the 2 of them. But the classical Delphi can have any number of rounds, but usually doesn't have an in-person meeting. So that's the difference between those terms. As I said before, though, the Delphi and modified Delphi are by far not the only ways of getting agreement from experts or engaging different experts. And these are all different things. And we'll talk about each of these in at least a little bit. So let's dig a little bit into what really makes a Delphi panel special. So the way I describe it is, a Delphi panel is a structured interaction that brings together multidisciplinary experts, uses quantitative methods to achieve consensus under conditions of uncertainty. So, structured and unstructured, what do I mean by that? Well, an unstructured on the right, and this is not, this taxonomy is of my own devising, so apologies if anybody thinks that they have a better one. Please let me know, and I'm happy to use it the next time. Unstructured are those discussions where you're just really letting people talk, and BOGSAT is an acronym for bunch of old guys sitting around and talking, which is kind of a joke, but not really, because that's how most guidance is developed, was clinical guidance anyway, was developed up until about 20, 25 years ago. But a little more structured than that is something called a nominal group process, where it's people sitting around and we have them share their opinion one after another, but they just share their opinion and they don't really interact. So it's a nominal group because it's a group in name only. There isn't that interactive process. So not really structure about how or what you do. NCCN guidelines are an interesting case. This is, many of you will be familiar with, this is the National Comprehensive Cancer Network guidelines that are widely followed in the field of oncology and hematology. And those are somewhat structured, and they are certainly more structured than people just sitting around and talking, and they do require a vote. But there is not really the kind of structure that you see with these methods further on the left. IDEA, Cook's Classical, Shelf, Delphi, and Modified Delphi. And the structure that those methods have is like what I described for you at the outset about that, the Modified Delphi. There are a sequence of steps that you have to go through. So IDEA stands for investigate, discuss, estimate, and aggregate. And so those are steps for coming up with like a probability estimate. And you have to follow those particular things. So that's one difference anyway between those different methods. Another thing is the use of quantitative methods. So by quantitative, I mean doing something that can be added, subtracted, multiplied, or divided. And so the qualitative methods, this nominal group, BOGSAT, and I would include NCCN guideline development here, because although the NCCN process involves a vote, that's not something it's kind of binary. You vote up or down on something. Whereas in these quantitative methods, you are asking people to give a numeric answer to something. Commonly, in many of these, you're asking people to estimate a probability or a likelihood or a number of events. But whatever it is you're asking, you do want some quantitative answer. And in our modified Delphi process, we ask, usually on a 1 to 9 scale, we ask how appropriate is this behavior? So the example that I gave you about sickle cell disease, the question we asked in that one was, how appropriate is it to schedule follow-up at this interval? Or tell us the number of weeks between visits that would be most appropriate. So there's a number there, but it isn't a probability. I also want to just kind of highlight, and I'll explain this in a second, that by quantitative, I don't mean the output has to be numeric. So as I said, in those IDEA, Shelf, Delphi method, you are looking for a numeric output. You're looking for, like I said, a probability or a number of things. In the modified Delphi method, although you're using a quantitative process, you can have as your output something much more conceptual, and we'll have some examples of that in a moment when I bring the panelists on. But this, to me, actually is the most powerful thing about the method, and to my knowledge, it's the only method that can be used to do expert engagement that is quantitative in this way, but can be used to answer much more complicated conceptual questions. Okay, but at this point, I want to bring in our panelists, Kathleen and Tiffany. So if I can have you join us. Great. Thanks for joining and helping me out today. First thing I want to do is just have you introduce yourselves a little. Tiffany, will you tell us who you are and what you do?
Sure. Hello, everyone. I'm Tiffany Kwok. I've been in drug development now for nearly 20 years, and I lead health economics and outcomes research at Crinetics.
Great. Thank you. And Kathleen?
Hi, I'm Kathleen Villa. I lead up the medical affairs evidence generation function at Ionis Pharmaceuticals.
Great. Thank you. And I've worked with both Tiffany and Kathleen on using the modified Delphi panel process. And so I thought they'd be good people to talk about what it's actually like from the client side to be involved in one of these. So Tiffany, you want to tell us a little bit about what the panel was about and what your experiences were?
Sure. So our panel was to estimate healthcare utilization in AL amyloidosis. And AL amyloidosis is a rare disease. We had a lot of challenges in terms of obtaining data, looking at the burden of disease, looking at the treatment patterns, and also understanding the utilization in terms of the staging of the disease. So when we had looked at claims data, we had difficulty there because we didn't have an ICD-10 code prior to 2017. And in trying to put together this story of what the unmet needs were, what patients were facing, we really had a lot of challenges. And that's why we decided on using this Delphi panel. So with the Delphi panel, we had a couple of objectives. We wanted to understand by the staging of the disease how the utilization differed, what types of labs were being used, chemotherapy, procedures, healthcare visits. And then we also wanted to understand institutional differences as well, whether or not practice patterns were different between, different institutions, versus a referral center versus a community practice. So we had 12 panelists on our panel. 11 of them were clinicians, and then we had one patient advocate who was the president of the patient advocacy group. So we learned a whole lot from this panel itself. And in addition to understanding the utilization, we also wanted to engage with our panelists. So it was an educational effort on both sides. We understood more from the sponsor side, and I think also our panelists also understood more about the differences in practice patterns and the utilization that patients had faced. And we also had panelists who went back to their charts and pulled random samples of patients to confirm what they thought was true or not true. And so in doing so, they recalibrated their ratings, which I thought was one of the key takeaways just from this exercise and the study alone. So it was very, very useful for us.
Yeah, great. That's a great summary. Thank you. And actually, I have here, I have the, let's see if I can find it. This table is kind of what we used. I'll say that the rating form in the case of the panel that Tiffany described looked a lot like this table, which is an output table in the paper that we ended up writing. And it's a little small, but what you can see is that in the columns, it describes periods of time and types of patients. And so that's what I mean when I say a patient scenario. So in this first column on the left, it says for 1L, first line, and we're asking about utilization over the first year of treatment for people in, as Tiffany said, different stages of disease. So stage 1 or 2 in the left-hand column, or stage 3 or 4 in the next. And then in each row, we ask a specific question. What's the likelihood of this, or how many of these labs do you think people would have, et cetera, et cetera. And so this is kind of, and the numbers that are shown here are the consensus numbers. Those eventually the estimate that we came to. But two things I also want to kind of highlight that you said, Tiffany. One was how much the panelists themselves enjoyed it. And this is really, I mean, I think it's kind of one of the secret strengths of the process is that the experts don't often get a chance to sit together for this uninterrupted time to talk about things like this. They go to conferences and they'll have meetings in hallways or hear a lecture, but this sort of discussion isn't that common among clinicians. And so they usually really enjoy it. They come away from it really kind of engaged and charged up about it. And the other thing you mentioned was that we had a patient advocate on the panel. And people always ask me about, so I've kind of been a big proponent of non-physicians on these panels. And people always ask about that. My experience has been that non-physicians contribute a huge amount to the panel. So I usually try to get advanced practitioners like nurses and physician assistants, as well as patient advocates. And even though a patient might not know the answers to the particular questions that we're asking, what I think it does is it changes the dynamic of the whole conversation, and it really recenters the patient. Doctors can sometimes go off on these tangents about very little things, and patients are really having somebody there who can bring it back and say, but my life is affected in this way, and that kind of recenters it. So I think that's really an important aspect. So great, Kathleen. Maybe you can say a little bit about what the panel that you worked on was about, and what you got out of it.
Yeah, thanks, Michael. So at Ionis, we have a product in development for a very rare condition that affects just a few people per million in the population. And so there isn't a good understanding of how to identify or diagnose these patients. And even among specialists in this therapeutic area, there isn't a good understanding of how to identify these patients. Or if you get someone in your practice by chance, maybe once or twice in your career, who has some of these characteristics, even understanding whether this person might actually have this condition. So we were interested in using this approach to bring together specialists and others, a broader group, as Michael just described, to talk about what are the main clinical characteristics that would help identify this rare patient population. So in our situation, we really wanted this consensus to be driven by the clinical community, and did not want our own involvement as the manufacturer of a treatment. Oh, and I didn't mention that there's no current treatments for this rare condition, which even makes it harder to identify, because in the past, these patients have not been treated. So we really wanted this consensus to come directly from the clinical community. So we came to PHAR and asked to help facilitate this approach in a double-blinded manner, where we would not know the participants, and they would not know that it was sponsored by Ionis. We did sponsor the project, but we weren't involved in the development of the project, or in the consensus discussions, or any of those aspects of the project. Although we did work with PHAR to have an understanding along the way of what the rating form looked like and what the evidence review looked like, in case we had particular expertise that we could lend along the way.
Yeah, that's great. And maybe I'll just show what the output of that panel was, just to give an idea. So,
yeah, and Michael, did you want me to go into a little more detail about how the process went, or did you want to describe that?
No, you go ahead. Go ahead.
Okay, so in the case of this kind of diagnostic score, there were a couple of different, it kind of, I think, had a qualitative and a quantitative component to the project to get to this risk scoring calculator type of approach that you see here. And the first was, as the PHAR team often does, I believe, in these modified Delphi panels, is do a rating form. And in this situation, there were many, many patient profiles that the panelists were asked to evaluate, and with many different clinical variables. And so they went through a consensus process to look at those clinical and patient characteristics, and to sort of weight the importance of those in identifying the disease. But after that consensus process, there was a sort of a analytical process in which their responses were incorporated into a regression analysis in order to come up with this sort of more quantitative weightings for each of these variables that ended up in the calculator.
Yeah, no, exactly. Thank you. That was a great summary of that. So I think, right, I mean, this is a very different example than the last one. So in what Tiffany did here, this is much more in line with the kind of result that you would get out of a traditional Delphi or some of those other methods that I kind of touched on, numbers and counts of things. But here with Kathleen, this risk calculator really was the result of a lot of thinking from these clinicians about how would we know, how would we estimate whether a person is likely to have this condition or not. And as Kathleen mentioned, when you're talking about a rare disease like this, there just aren't thousands of patients with data around that we can aggregate and analyze. And so using these patient scenarios, which and a scenario in this study consisted of kind of each of the individual variables. So you can imagine that one scenario was a patient aged 1 through 9 who had a body mass index of less than the 85th percentile, who had symptoms but no history of this condition A, and had no other factors that could contribute to abnormal lab tests, and had the lab result of less than 2 units per liter, or whatever the thing is here. And so each scenario was something that specific, and the panelists then rated how likely they thought a person with those characteristics would be to have the condition. And so we used that information to weight, to create the weighting that you see here using regression modeling. And we actually did even a further step. Luckily, we found a registry where we were able to collect individual patient records and look at the whether patients with meeting these characteristics did in fact have the condition of interest. So we were able to do some validation as well. But all began with the expert panel. And you mentioned a couple of things that I think people in the audience will be interested in, because I've heard them come up before. And the first one is the blinding process. So I'll just say a word or two about that and then ask you kind of about your experience. I came up with this idea of blinding these panels maybe 15 years ago. So when I started doing it, my experience had been from the academic side, organizing these things. But once I got into industry, what I quickly realized was that when you go to publish the results of an expert panel, where you as the company have funded it and have done and have paid every single panelist, readers of that publication are going to be suspicious that you asked for a particular outcome and you got what you asked for. And so we came up with this idea that we could blind it, as Kathleen just described. What we do is we'll often begin with a list of names that is maybe the company provides us. But that's the last they hear of who the panelists are. So they'll give us some ideas and we'll find a couple people and we'll branch out from there to find more people. And then the members of the panel know that that there is a funder of this, but they don't know who. And the company knows that we are finding these panelists, but don't know who they are. And we go through the entire process like that, with neither side knowing the other side. When it comes time to, most of these things end up, as you can imagine, in a publication. And so when it comes time to do that publication, we write a draft of the publication. And then before it gets submitted, we unblind the panelists, because they need to be able to make their disclosures. And you can't disclose if you don't know who the person is. So we unblind it then. But it's after all of the analysis, all of everything has happened. And we've had really good luck with getting, or that's not luck, I would say, but we've had good success at getting those things published if you do it that way. Even unblinded panels, we get published. The one that Tiffany did was not blinded, and those can be published too. The key is that you have to be able to make the case that in an unblinded setting, that whoever paid it wouldn't have influenced what was going on. So, Tiffany's company didn't have any particular interest in a particular number being the outcome. So blinding wasn't really relevant there. So yeah, that was an important aspect of that. Kathleen, anything else you want to say about that process?
I wanted to.
Blinding part, I'm sorry, I wasn't very specific, but I meant just about the blinding. Was it scary not to know who the people were, or what was the?
I mean, we did go into this project knowing there was a certain level of risk of not being in control. I think that's anytime you sponsor a project where you're not going to be involved, you don't know what you're going to end up with. So there was a certain amount of that type of hesitancy, but ultimately that was our primary objective, was for the clinical community to come up with their own ideas about. I mean, they are the experts, and we wanted them to be able to come up with their own ideas about how to identify these patients. But I did want to highlight one thing you said, Michael, which was the idea that the experts do, I think to a certain extent, it's hard work for them, but on the other hand, I think they do enjoy coming together with their other, experts in their field to discuss these issues, which they don't have a lot of opportunities to do that. So, we've heard anecdotally after this process was unblinded that the KOLs really appreciated the opportunity to get together to talk about to what is to them a very important topic about these patients and identifying them so that in the future they would be able to treat them.
Yeah, no, that's exactly what my experience has been too. That I think because the questions we come up with to ask for a panel like this, the reason we're asking a question that you as a company have this question is because there's some information out there on it. So there's some scientific evidence, but the answer isn't a slam dunk. If there's 3 randomized trials on something, then we don't do a panel on it. So what you get is a situation where you're in a place where experts have information, they've processed it, they've thought about it, and they have some ideas. But those are kind of personal ideas usually. They haven't really been widely shared with other people. And so they get the opportunity to share those. And that, I think, leads right into one of the key, there's some magic in this process. And one of the elements of magic, I think, is the way that it helps people, that it reduces bias. So what happens is when somebody makes a statement in a vacuum, they're like seeing patients and they say, this is what I'm going to do. And there's nobody there to argue with them. And they do it and it works out well, and they become convinced that that's the best way to do it, given the evidence. But somebody else does something different and they have a different experience. They're also a successful experience. And those two people are operating kind of independently. But you bring them together and you hear your colleague that you respect or admire saying, no, I do it this way, and I've had good results, or this is how I think about this problem, and this is how I solve this clinical issue. It really changes people's mindset in a way that kind of just hearing a lecture or reading a paper doesn't do. And so I think hearing other clinicians talk about how they do what they do or what they think, how often, and Tiffany, in your example, as you said, somebody came to that meeting, with information like, I looked at my patient records and this is what we did. And I think that is really powerful, and it helps move people from the position they came in on. And so, in that little cartoon that I showed that first round ratings, and then this discussion and second round ratings. And people sometimes ask, like, how much change do you expect from one session, one round of ratings to the other? And it varies widely, but it always drives toward consensus. People always come closer to agreement after talking through. There's always places where somebody will say, yeah, no, after hearing that, this is now what I believe something different. So that's one of the elements of magic, is that reduction of bias by hearing other people's view. The other thing is this mental process that people go through when they're panelists in one of these panels. So as physicians are in training, they're taught to break problems down into these kind of very granular level. As a medical student, you kind of learn, create this differential diagnosis of 30 things this could be, and think about the characteristics of each of those diseases and what this patient has and what they're most likely to have. But as you can imagine, experienced clinicians don't think like that at all. They do things very intuitively. Oh, this patient is about like this. Okay, I'm going to do this. And so they've sort of aggregated all their clinical experience and knowledge and everything they've read into this very fast thinking approach. And there was a book out a few years back called Thinking Fast, Thinking Slow. I think that described this very, very well. There are almost two different brain systems that are involved in this. And so when you do those intuitive judgments, you're using this quick thinking. And it's not that it's bad to do that, but that's what experienced people do. That's what experience is all about. But what we do is we take these experts who are used to now using this quick thinking and we force them to slow down. So that's what that rating form does. It creates this very detailed grid of things. And so they have to think, this question now I'm answering about a patient who's only a little bit different from that patient. They're only 5 years older. They only have one more risk factor. How are those two answers different? And so you're slowing down their thinking. And when they come together in the meeting, each of them has been through that experience of being forced to slow down. And so they have kind of interrogated their own process a little bit better. And so when they come and sit down and talk about it, what they'll say is things like, well, I thought that this was, really important, but I saw that my answers weren't very different from one thing to the other. So they intuitively felt like something should matter, but then it didn't really matter to them in the end. So those are the two kind of things, that reduction in bias from hearing other people's opinions and the slowing down, that I think really kind of help us get to the best possible outcome. One of the questions that came in was about how to find experts. So maybe let's talk about that a little bit. Tiffany, do you want to talk about, since you had some input into this, you weren't blinded to it. So can you talk about that process, how that worked?
Sure. So we already knew several of the panelists that we wanted to invite for sure. And they were panelists that were on our trial or panelists that we wanted to do outreach that were not a part of our trial. So these were definitely physicians that were highly tiered, very visible, but were not enrollers within our trial. We wanted to reach out to them. And then we also had a chair that we were working with. And so in working with this particular chair, we were able to pinpoint, the different panelists and knowing that there would be differences in opinions. We just wanted to make sure that we had a diverse set of opinions within our panelists. So those were the criteria that we were looking for. So, folks who were on our trial and then folks who were not on our trial that we wanted to do outreach, and then a diverse panel of different physicians. And at the same time, I do want to add, none of these panelists had any experience with any type of healthcare utilization or health economics type of project. So I think the majority of them, Michael, when we approached them, they had said, well, this is not my usual study. I'm not sure how much I can contribute, but at the same time, this seems like a neat and fun study. So let's do this. I think that was the very common response that we'd gotten from our panelists, and they ended up having a really, really good time together with this discussion with both of the panels that we had.
Yeah, great. Yeah, and I think that's important that you talked about identifying the chair, and we usually do try to do that first. We try to identify one person who then we can branch out from and ask. And that chair has a few more responsibilities for the panel. They're not the moderator. All of the panels that we use are professionally moderated by me or one of my colleagues who've done this many times. But they are involved a little bit more. And one of the people asked in the chat about compensation, and we do compensate the panelists. And in our panels, most of the people are clinicians. So they're compensated at a clinically appropriate rate. Usually the company has some fair market value that they're compensated at. Another one of the questions was about people dropping out between the rounds. We don't, so in our process, in this modified Delphi process, remember there are only 2 rounds with this in-person meeting between them. So we don't really see drop off between the rounds. Our ideal number of participants in one of these panels is like 10 or 11. So we usually recruit 9 to 12 or maybe 15 people altogether. And usually somebody will get sick or have some childcare crisis or something like that, and they won't be able to make it. So we'll lose 1 or 2 people before we get to the panel. But often those people can stay involved in the process and even do ratings, although it's a little bit, it depends on how the discussion goes, whether those ratings are useful. The other thing that came up, somebody asked this, was about protecting personal information. And our panelists are not being asked, so they're not patients most of the time, or even if they are patients, they're not talking about their personal experience. So the patient advocate that's on this, that we will have on a panel like this, may be a patient, but they're also usually involved in some advocacy. And so what they're talking about is the patient perspective. They're not describing their protected health information. So these studies are not human subjects, studies by definition. And you can get a waiver of, human subjects review. At least that's been our experience, that it's easy to do that.
So I wanted to add something about this concern about dropout. I think having the virtual panel made it a lot easier for people to sign up because there wasn't travel, right? So it wasn't live. And so I thought that really reduced and minimized any issues with dropout.
Mm-hmm. Yeah, that's a great point. So when that pandemic hit, I'd never done a virtual panel before, and we had one scheduled. I think it was like scheduled for April of 2020 or something like that. And so we went into it with a lot of trepidation about how that was going to work. I mean, so much of the panel is, this kind of back and forth. And what we found was there actually was an advantage to the virtual, and that is that it's easier to schedule. So instead of doing one solid 8-hour block, we do 2 4-hour blocks or 3-hour blocks or something like that, and no travel. And so panelists are actually it's actually easier to get it on the books, and there are advantages to it. I would say that. And it also turns out that when I talked a little bit at the beginning about that nominal group process, so that's one of the techniques that we lean on to moderate these panels. So what I do is I ask when we get to a question where there's disagreement, I ask one panelist to describe how they came to their answer, what was behind their answer. And after they give it, I kind of summarize it, and then I turn to the next panelist and ask how they did theirs. So I don't really invite them to argue with each other about what their difference of opinion is. I just try to make sure that they're sharing it. And so I think because that the moderator is in the middle of it, that Zoom meeting actually turns out okay, because it isn't just a lot of crosstalk of people trying to get into each other. I think if it wasn't moderated in the way that it was, it would be much harder to do those virtual things. But we did, and actually it's in the handout section. We did a paper just describing our experience with virtual versus in person. And it's really hard there's so much difference from panel to panel. I mean, and we have a limited experience, so the N is small. But honestly, I don't see that much difference. The one thing that I do note is that the informal interaction that panelists have with each other in the in-betweens is nice to watch. So people connect with each other in a way that, oh, you're doing this, I'm working over here, I do this, like, oh, we should talk about that. So there's that sort of informal connection, and that doesn't happen when you're doing it virtually. But I think otherwise, it works really, really well.
It does. And ours was during the pandemic. And actually, I was a little bit concerned. I remember telling you that I was a little bit concerned because of the 12 panelists. We had quite a number of strong opinions, but I thought that you did a really great job in terms of moderating. And, for any of the audience members who are concerned about being on the sponsor side and sitting in the peanut gallery, I don't think the panelists even noticed that I was participating after a while, because I didn't say anything.
Yeah.
I just really just sat there and listened.
Yeah. Yeah. And usually our sponsors have a number of different ways of being involved in the panels, and sometimes we usually try to get, if we're in person, we try not to have the sponsor at the table. We sit around a conference table. Sometimes they're in the room off to the side, usually not talking much. Sometimes sponsors will want to record these things. Sometimes just get the report or listen in. So people have different opinions about how to do it. So we have a few minutes left. So I want to do something. Let me just take a look here at this question. So there's some questions about different kinds of panels and what one can do with them. So let me just go to a couple of things here that I have examples of and just kind of highlight a few things. So this is another panel that we did. This is a panel on the Grail, which is a company with a diagnostic test. Wanted to know about the likelihood of people moving from one stage of cancer to another over a given period of years. So their idea is to find cancer early when it can be cured. And so some of this, as you can imagine, in the field of oncology, you don't just let people go from stage 1 to stage 3 cancer. So there's no good way in using data to know how long it takes to get from one stage to another. So we use an expert panel to come up with these numbers, how many years. So that's sort of like in the traditional mode of getting a quantitative output. This is a flow chart of how to taper a particular medication when people have this immune thrombocytopenia. They're on a particular drug, and what happens when they're getting better? Can you taper the drug off? And so we use an expert panel to come up with guidance for clinicians on how to stop these drugs. And you can see this is kind of the main output, was this flow chart. But on the right-hand side, these color-coded boxes, these boxes are really are coded to indicate areas of agreement or disagreement, or when panelists thought it was inappropriate or appropriate to do a particular intervention. And so that came directly from the numeric ratings. So we had these ratings. If people gave a low score and said something was inappropriate, we colored it red. So you shouldn't taper the drug when people are responding but still have a low platelet count. That would be inappropriate. But then down here at the bottom, you see cases where it would be appropriate to taper. So that's another kind of use. This is a classification system, severity classification system for sickle cell disease. And that's like, how severe is your disease, class 1, class 2, or class 3? That was a system that didn't exist until we did this panel, and we used experts to kind of come together and decide how the disease should be classified. This was a panel on managing adverse events of treatment. So that's yet another way it can be used. So just to kind of get your audience's mind rolling about different things you can do. Let's see. Question came in about, do you encounter some sort of conformity bias during the discussions? So it's a good question. I mean, I think my answer is I'm not sure it's bias. I mean, yes, I think people do listen to each other and change their answers in response. But these people the people that we're using for this are all experienced in their field. They're experts in some way or another. So I don't think they are as inclined to just give an answer that is, with the group. So they do maintain, and we do see at the end of these panels, it's not atypical to still have 10 or 15% of the items still with disagreement. And the way we handle that in a publication is we just say, here are the areas of agreement, and this is the areas where maybe further research is necessary. People disagree often because there's just very little information, and people are just using their own, experience to guide them. Here's another one. Beyond publishing in peer-reviewed journals, how do you demonstrate that? How do you measure the impact of results to clients. Yeah, so maybe the two of you could speak to kind of how useful these panels were to you, what you got out of it.
I mean, I think the way you would measure the impact would differ based on the objective and the type of project. So in my case, where we were looking at, we asked the panel to look at kind of diagnostic criteria. I guess it will be a couple of things. One, the extent to which the clinical community embraces this type of scoring system.
Mm-hmm.
And we know that it will be presented at this key therapeutic area meeting and then published in a peer-reviewed journal, which is in process now. But also, if the community really embraces this approach, then we'll likely see it being used and recommended in treatment guidelines and talked about more broadly in this kind of clinical community and sphere for this patient population.
Yeah, and we've seen the outcome of these panels be incorporated into actual clinical practice. I mean, one we did was to develop an order set for the emergency room for a particular condition, and that got used. But I will say, actually, Kathleen, one of the reasons that the panel that we did with you, one of the reasons I think it's going to likely have impact is because it didn't end just with that panel. We didn't just produce this and publish it. We did these other activities that we talked about including validating it to some extent. And so, so I think that sometimes it requires some add-on activity. The original panel work that was done at Rand and UCLA, it had a really dramatic impact because clinicians were fed, at least the project that I worked on in the late 90s was the appropriateness of hysterectomy. And we found lots of inappropriate hysterectomy and fed that back to the practices where we'd collect the data from. And so they changed some of their practices. So it depends on what you do kind of in the ancillary setting. And Tiffany, for you, what about for yours? Yours was a much more discrete outcome that we were looking for there, but what about the impact for you?
Right, ours was a discrete outcome, and at the same time we wanted to use it for our models. And then also at the same time, our chair of the panel actually came back and said, no, this is really, really interesting work, and it would be great to develop medical education, not just off of this particular publication, but from our bundle of publications that we had done. And we also had a registry as well. So
yeah,
I really see this as one tool in your armamentarium of many different tools. And there's different strategies to engage with your stakeholders. And this is just one of those very key, impactful ways to do so.
Absolutely. Yeah, thank you. And actually I see that we're just about out of time. So that's probably a good point to just kind of wrap it up. But I think what you said is a perfect wrap up, that this is this is one tool. I think it can be very useful in certain settings, not everyone. I mean, I think in areas, as I think I alluded to at the beginning, in areas where there are multiple randomized trials, to answer the question, it's not particularly useful. And when there really is no information, it's not particularly useful because then people are just going on their opinion. And so there's nothing, much to anchor to and no way to get them to change. But in that in-between space, it can be very, very useful. Well, I want to thank Kathleen and Tiffany for taking the time to join us. I really appreciate your input. I think it was really helpful to have that. For all of the audience who's still out there, check out the handouts there. I put publications related to probably maybe half a dozen related to the Delphi panel, and then another bunch related to some of those other methods that I touched on and we didn't really talk about in detail. But if you have additional questions that we didn't get to, feel free to leave them in the chat and I will try to answer them. This meeting itself will be posted, so you can watch a recording of it. And over the next day or two, I'll try to answer questions that I haven't answered yet, and we'll be posting those answers as well. So thank you again, everybody, and enjoy the rest of your day.
In closing, we hope you enjoyed this HealthEconomics.com webinar sponsored by Partnership for Health Analytic Research, and I hope you enjoy the rest of your day.